Hello, everyone, and welcome to today's webinar organized by Working Group 1 of Cost Action Sustain. We are pleased to have two excellent speakers with us today, Professor Muhammad Saqib from Institute of Soil Environmental Sciences, University of Agriculture, Faisalabad, Pakistan, and Dr. Leila Jahanbazi, a postdoctoral researcher in the Department of Agroecology at Aarhus University, Denmark, who will share their research on the soil salinity monitoring and management approach. Please feel free to use the chat for questions. We will have a short discussion after each talk. With that, I would like to invite Professor Muhammad Saqib to deliver his presentation titled Unraveling the Interplay of Co-Occurring Stresses in Salt-Affected Soils, a Comprehensive Analysis. Dr. Saqib, the floor is yours.
Okay, thank you very much, Dr. Ishaq, for this nice introduction, and I'm also very much thankful to the Sustained Cost Action Program for this invitation and giving me this opportunity today to present my work on salt-affected soils. Let's start with the presentation. I will try to share my presentation. Okay, is it visible?
Yes, great.
Okay. So I am Professor Dr. Muhammad Saqib, and from the moment working at the Institute of Soil and Environmental Sciences, University of Agriculture, Faisalabad, Pakistan. I am a fellow of Alexander for Humboldt Foundation, Germany, and also fellow of German Academic Exchange Service, which is DRD, Germany. I worked at Justus Liebig University, Germany, in Gießen for both these fellowships. Then I had the chance to work at the University of Tokyo, Japan, as a fellow of Japan Society for the Promotion of Science. Then International Foundation of Science from Sweden, they actually funded my work here in Pakistan. I am a life member of Science and Society of Pakistan, and I had also the chance to work at York University, United Kingdom. These are the contents of my today's presentation. I will cover salt-affected soils in general, their extent and importance, then how the plants they grow on salt-affected conditions, what are the effects, how they respond, and what are the improvements that can be done. Then, of course, mainly I will come to the co-occurring stresses under salt-affected conditions, which include sodicity, drought, waterlogging, and soil compaction particularly. And then we will discuss that what could be the way forward for the salt-affected conditions. I will present one of my case studies and some general recommendations. So today we are discussing salt-affected soils. So why? Because they are the main reason of land degradation around the world. About 75 countries of the world, they are affected by salinity to some extent. That covers about 7% of the world land and one-third of the irrigated land of the world. And these soils, these are vital for the food security and climate change resilience in these conditions, because these are the soils, these are the lands which are inhabited, which are having the people who are living there under very poor conditions. So it is not possible without using these degraded land resources to actually make food security possible and climate change resilience possible in these conditions or for these areas and regions. And we in Pakistan, we are worst hit by this land degradation due to salinity and climate change induced environmental stresses. Like recently, we are actually facing the floods and sometimes droughts also. And over time, as these graphs show, the conditions they are actually expected to get worse with time. as we all know that we as a global citizen, we are actually trying to fulfill our responsibilities with respect to the sustainable development goals. And if we look at the sustainable development goals, soil, it is at the center of the realization of these goals. We cannot realize these goals without having sustainable use of soil, without you can say neutralization of land degradation, without stopping land degradation, and without rehabilitation of the degraded land resources. So it's very important that we study these, we use these, and we actually properly have production from these lands. These are graphs which simply show that if we are not using these sites, we are not actually improving these sites, which we will say as cost of no action. So that is increasing with time. So we need to actually intervene these sites even if we have to invest something. Now, if I just look at the characters of the salt factor sites, their classification. So, salt factor sites are generally classified as saline sites, sodic sites, and saline sodic sites. Saline sites are more in area, and generally salt factor sites are considered to be saline sites by many experts also, general people also, you can say. So saline sites, they only have high soluble salts. So means that they have high electrical conductivity. The sodic and alkali sites, they have high exchangeable sodium concentration in the soil, which actually affect the plant growth. And saline sodic sites are alkali sites. They are having high soluble salts as well as high exchangeable sodium. So in most of the salt factor sites, if only soluble salts there is problem, in many of the salt factor sites, additional sodicity. And later I will show that code how the additional sodicity that additionally affect the plants under salt factor conditions or under saline conditions. Now, nature has blessed us with the diversity in the salt tolerance of the plant species, ranging from very salt sensitive to the highly tolerant plants. We need to actually look at the characteristics of these plants and to utilize these plants. Presently, if we look, 90% of the human food it comes from only 30 plant species and 50% from just 4, which are rice, maize, wheat, and potato. Now, the salt sensitivity of our staple food crops will make them redundant in very near future. It's based on the data, based on the you can say present conditions, and that will leave the space for the salt tolerant plants as the future crop, future food crops, and crop for many uses. This picture shows a general look of the salt factor soils, where we can see that plants they are growing, but their growth is not very good. Their yield will not be very good. The part of the plot that is highly saline, there is no you can say growth of the plant. So usually in patches we can find crops over there. We can find the different grasses, and this is general you can say picture of the soils. So they are characterized by low crop yields and vegetation cover, high poverty and malnutrition. So people are living there, but usually they are poor people and malnutrition people. Then environmental pollution and land degradation, that is there in these areas. And the common co-occurring stresses in the salt factor soils they include drought, waterlogging, soil compaction, heavy metals, air pollution, and high low temperatures. Now, the way forward for these sites is reclamation and rehabilitation of the salt factor site using certain amendments in different ways. And the other one is the selection, development, and characterization of the salt tolerant plants or salt resistant plants. This is just a view of the reclamation which we usually practice, that for the saline soils, we are simply removing the salts, leveling the soil, and then evaluating the soil for its extent and its intensity of the salinity, and then simply giving heavy irrigation if it is saline soils, and then the salts they are actually leached down, and we can use it for crop, for rice, for wheat crop. Similarly, if the soil is sodic or saline sodic, then we need to add gypsum or sulfuric acid in the soil, so that the sodium on the exchange sites that can be removed and the soil becomes good quality soil. Now, before going to the detail that how different other stresses they are actually affecting plant growth and soils, just in the beginning, I would like to actually discuss some of my experiments with respect to the salinity and plant growth. So these are from the wheat. So as we know that salinity, it reduces plant growth, it reduces heat of plant, and these effects are of course after having the effects at the physiological level, at the molecular level, and particularly the sodium concentration that is in, you can say, the center. It is increased under salt water conditions, that leads to low growth and low heat under these conditions. So in one of these our studies, we saw that the relative expression of antiporters, sodium to proton antiporters, that is actually playing the important role in the tolerance or in the sensitivity of wheat genotype. The genotype which was having a higher expression, it was actually accumulating less sodium in its particle tissues as compared to the genotype which was having less expression. So this at the molecular level is very important role. Similarly, certain other ions, particularly for example silicon, iron, zinc, boron, they have very important interactions with sodium. So in next our study, it was shown that the presence of silicon, it actually reduces the uptake and the accumulation of the sodium in plant shoots, and it relieves the plant from the saline stress. Similarly, under the conditions where higher iron and zinc was present, the plant growth was better, the genotypes which are having better potential to uptake these ions, they were better performing in the starter. So under salinity, the role of the membrane proteins would play important role in the uptake of the sodium, in the uptake of the potassium, in the uptake of different other ions. That is very important, and the uptake as well as the availability of the other ions like iron, zinc, boron, silicon, so that is very important. And the effect of other stresses on the salinity and on the plant performance under salt water conditions, that is also indirectly related to these membrane proteins as well as to the uptake and availability of these nutrients from the plants. Uh, this experiment is related to the effect of sodicity, presence of the sodicity under the saline conditions. So it's again on wheat, different wheat genotypes which were compared under saline condition and saline soda condition. So genotypes, they differed with respect to their response to salinity and their response to sodicity. Some have the similar response under saline and saline soda conditions, some have different responses, and usually this response was actually related to the uptake of the sodium. Genotypes having more sodium, they were actually less, you can say, tolerant, and the caloride concentration, that was less in plants under saline sodium conditions than the saline conditions. So sodium was more taken up under sodic conditions and saline sodic conditions, whereas caloride was higher under the saline conditions. So it means that the presence of sodicity under saline conditions, that affects the plant growth, and in most of the cases there is additive effect. So This is the response of the quinoa to saline and saline soda conditions. Because quinoa, it is a highly salt tolerant crop, but here we have seen that its response to salinity and to sodicity, you can say, or saline plus sodicity, that was different. Under saline sodic conditions, its growth and its yield, that was significantly more affected as compared to the salinity conditions only. And again, it was due to higher food sodium, low food potassium, and decreased membrane stability index under the sodic conditions. So plants do differ with respect to their response to salinity and sodicity and saline sodic conditions. Usually, the presence of additionally sodicity under saline conditions can have an additive effect on plant growth. This is very interesting study which was published in Plant Physiology and Biochemistry. Again, it's on wheat, and here we actually, under controlled conditions, try to relate the low calcium, because under sodic conditions, usually sodium is higher and the availability of calcium, that is low. So we try to study that how the response of the wheat genotypes, which were actually performing different under sodic conditions in the field, that is related to low calcium. So, as it is shown in this graph, the low calcium, under low calcium conditions, the response was similar as it was under saline sodic conditions, and the genotypes which are performing better under sodic conditions or under salinity and low calcium conditions, they were actually having better ability to acidify their rhizosphere, because rhizosphere acidification, that leads to better performance of the plants, particularly under sodic conditions and saline conditions. And similar results they were found in some tree species, particularly the acacia. Now, of course, as the literature says, and many of the studies, we see that sodium, exchangeable sodium absorption ratio under sodic conditions, that is considered as the most detrimental, you could say, from the plant growth from the soil. But some of the studies, they show that along with this sodium, the magnesium concentration, that is also important. So, in the literature, the exchangeable, it has been suggested that the exchangeable magnesium percentage, along with the exchangeable sodium percentage, that is also important for constitution for plant growth under these conditions, for sulfur retention, particularly these conditions where magnesium is also present in soil. Now, coming to the presence of the drought under saline conditions, as you know, the availability of the water is already affected under saline conditions, and if there is drought or there are water deficient conditions, of course, the availability of the water, that will be compromised. And in this study on wheat, we have seen that under saline conditions, when we, for 21 days, we restricted the supply of water to these plants, and we restricted that supply of water at different stages. So the effect of salinity, that was higher. So combined effect of salinity and drought, that was higher on grain yield as well as on growth of two wheat genotypes. So drought, again, it has a detrimental effect under saline conditions as compared to its effect under non-saline conditions, and it was evident when the drought was actually applied on all the important stages like pulving, fruiting, bloom stages of the field. So it affected, it reduced the yield, and again here the sodium and potassium, that was playing very important role. The sodium concentration, that was increased under these combined stress, whereas the concentration of potassium, that was decreased as compared to the individual stresses. So when the salinity and drought, they are combined, they have more detrimental effect on wheat growth and saline as compared to its individual effect, and that was due to its effect on the ionic composition as well as on the water relations. And here we have also further seen that the low grain number and weight in wheat and maize under salinity, water and wheat stresses, they may be due to reduced sugar transport to the developing ovaries, due to reduced activities of invertase and sucrose synthase in these conditions. Now, these effects we have seen not only on wheat but also another plants also, and particularly in jatropha and in guava. So again, the additionally present water stress or drought that was having a detrimental additive effect on the plant growth. So the stress tolerance of the plant that was decreased due to the presence of these conditions. Then another stress, which is waterlogging, which is very detrimental, particularly under saline conditions. So again, in one of our studies on wheat, we have seen that combined effect of salinity and waterlogging on the wheat growth and yield that was significantly higher than the individual effects of waterlogging or the salinity. And it was again due to increased sodium concentration under saline waterlogged conditions. Because under waterlogged conditions, usually the availability of oxygen that is affected, that is reduced. So as a result, the respiration becomes anaerobic and the energy production is reduced, and that leads to its effect on the ion exclusion, and that leads to higher sodium concentrations and lower potassium concentrations. And another important effect is on the nodal to seminar root ratio, respirate ratio, because under waterlogged conditions, the development of the nodal root that is very important for the survival of the plant, for the growth of the plant. So under saline waterlogged conditions, this was affected in the wheat, and that led to the reduced growth in these conditions. Similarly, the formation of parenchyma, particularly in the nodal roots, that is very important for survival under saline waterlogged conditions. And under these conditions, this was reduced. Parenchyma development was reduced in those waterlogged conditions as compared to waterlogging alone, and that also led to the reduced growth and yield of the this study shows the effect of the waterlogging which was created at different growth stages of the plant. Again, at different stages, stabilization, fruiting stage, grain stage. So at all these stages, it has detrimental effect. The combined effect was higher than the alone effect of the salinity at the waterlogging. But the occurrence of waterlogging at grain filling stage, that was most detrimental as compared to the stages. And again, it was related to the sodium concentration and potassium sodium ratio of the feed. So it increased basically the sodium concentration and decreased the potassium sodium ratio. Another stress which is particularly due to the use of machinery these days for the cultivation of the land, that is the compaction. So again, in this study, we have seen that the compaction that increases the effect of salinity on plant growth, and this is again a study on the wheat that when the soil was compacted, saline soil was compacted, it has more detrimental effect as compared to this effect when it was not compacted. However, when the soil was to some extent waterlogged, also then the effect of the compaction that was reduced, and that is understandable. But if we just look at the salinity alone, then the compaction that increases the effect of the salinity. So we we have seen that different common stresses, particularly drought, waterlogging, compaction, sodicity, so they all increase the effect of salinity on the plant growth and heat due to different reasons, including the physiological reasons, molecular reasons, and of course the reasons which are related to the root growth, which are related to the characteristics of the soil or soil properties. Now, of course, as we in the beginning we discussed and we said that it's very important to use these science. Now, how can we actually, or what are the problems particularly? So, if we look at the reclamation, so challenges and roadblocks for reclamation approach, so they include they require the good internal soil training. In most of the cases, we don't have the good soil training. Land leveling, which is in most of the cases we need to do the land leveling. Extra cost is there for that. Availability of good quality water, that is one of the most important, you can say, requirements. And most of the time that good quality water is not available in these areas. And we, for leaching, we also need to deep groundwater. Sometimes it's not because water line is there. So reclamation approach is prevalent, is actually available, but in many cases it's not possible because of these problems of these prerequisites to be met. So nowadays the ecological rehabilitation of saline and site, that is becoming more important through selection and improvement of salt tolerant plant resources. And here I am sharing one of case studies where we actually rehabilitated sulfate land at one of our farms through interventions through salt tolerant plant species, crop genotypes, and grass species, tree species. So a number of different plants we tried here, and we were successful to rehabilitate the soil. This is the upper view, that is the field view before the intervention, and the lower view is the field view after the intervention. Now it's almost around more than 10 years, and we are growing crops as well as grasses and grass trees are there. So for multi-purpose, we are using this site, and particularly at the moment, we are trying to actually demonstrate that this site can be used for upping the carbon credits also, because similar sites are available at other places. So if we can demonstrate that, we can use those sites for that purpose. So this is just a characterization of different plants with respect to their growth under different conditions. So under, for example, saline under drought condition, under waterlogged condition, under sodic conditions, which plants perform better and which plants they are actually having poor growth. So that under certain conditions, proper plants they can be actually advised. These are some general recommendations with respect to the ecological rehabilitation of the soil fertile land, use of these site-specific land use and management practices. We cannot use a blanket approach for maybe all soil fertile sites. We need to characterize the soil properly, specifically for a particular piece of land. We need to devise the management practices and uses. Taking into consideration allied stresses like drought, waterlogging, and heat, sodicity, compaction. So it's very important that when we are actually planning something, we need to know that what other stresses are there so that we can tackle those stresses right from the beginning. Raised bed plantation, that's very useful under these conditions. Drainage in waterlogged areas, like for example pumping water, also other ways. It's important. Selection and development of stress tolerant crop varieties and domestication of wild plants. Of course, this is a continuous process, and we need to do that to utilize these sites. GIS and remote sensing based monitoring of the existing and periodic changes in the soil and water stabilization. Develop science and technology parks for the technology transfer for actually using the knowledge which we actually generate from the local research stations for the farmers, for the end users. So with this, I would like to acknowledge all my collaborators from around the world, my students, funding agencies who have really contributed to this work. And thank you very much.
Yeah, thank you very much, Professor Saqib, for your insightful and comprehensive talk. And now we have a very short time for questions, and we have already one question from Islam Hassan. Hi, Professor Muhammad. In the light of your expertise, what's the most affordable technique to boost plant resilience against the salinity stress? If we can shortly answer, because we have another presenter.
Yes, I think it depends again on the specific conditions of the soil. So if it is only saline soils, maybe by heavy irrigation, we can decrease the salinity at that particular place. If, for example, sodic conditions are there, we can add gypsum over there, and alongside that, we can use salt tolerant genotype. For example, I have shown here most of the work from the wheat. We have different wheat genotypes having different salinity tolerant. So we can use plants which are salt tolerant. So we can have a combined approach, reclamation you can say, or addition of certain elements. Similarly, if nutrition is a problem, we can actually add maybe more fertilizer or certain other elements. So in this way, we can have better plant growth under scientific conditions.
Thank you, Professor. If you can shortly answer Lutfi's question, what do you think about date palm for saline soil?
Uh, I think it should be successful again with certain interventions, depending on the specific soil conditions where we are actually growing. It's very important. So we cannot have a generalized recommendation. So we need to actually analyze the soil, that whether it is sodic, whether it is saline, and again the soil texture, which is, of course, it was not in the preview of this topic, so I did not actually touch that. But it's also very important to look at that what is the texture of the soil, what is the water available. So we can, of course, use wave form, but keeping in view the soil characteristics, water characteristics, we need to adopt certain management practices.
Okay, thank you, dear Professor Saqib. And now you can stop your sharing. With that, I would like to invite Dr. Leila Jahanbazi to share her presentation titled Soil Salinity Digital Mapping Using Random Forest Model. Dr. Leila?
Yeah, do you hear me?
Yes, we can hear
Okay.
you.
And now
Yeah,
I'm sharing my screen.
perfect. Yeah, you can
Yeah,
go for slideshow.
good. So good afternoon, everyone. Thank you for your participating today. I'm pleased to join you today for this webinar. My name is Leila Jahanbazi. I'm currently a postdoctoral fellow at Aarhus University in Denmark, which my research is primarily focused on climate change and its impact, specifically in terms of greenhouse gas emission. I completed my PhD in soil science, and during which I concentrated on the mapping of soil salinity in field scale. And today I will presenting an overview of this work and sharing some of the key insight and finding from my research. Thank you for your attention, and I look forward to our discussion as well. So soil salinity is defined as the accumulation of soluble salts in the soil, typically measured by the electrical conductivity of the saturated extracted. A soil is considered saline when the EC exceeds then 4 decimeter, a level at which plant growth and function can be seriously affected.
Dr. Leila, sorry to interrupt. Could you please switch to full screen slide?
Because for me, it's now full screen. Let me try this one.
Yeah, now it's good.
Is it okay now?
Yeah, it's okay.
Good. So salinity negatively impacts overall soil health by damaging soil structure, reducing water infiltration, and increasing erosion. It also leads to lower level of organic carbon and also essential nutrient, reducing biodiversity, and therefore low crop yield. It could be natural due to saline parent material like the study area that I was studying in arid region in Iran, and also it could be because of anthropogenic action like unsuitable soil and water management.
We can't see your picture. Your camera is closed, I think. The lid is closed, or what?
But you can see my presentation, true?
Yeah.
I think let's keep it like that if there's a technical issue.
Okay.
So in recent decade, the rapid growth of the global population has made saline agriculture increasingly important for ensuring food security. It estimated that world population increased to 9.8 billion by 2050, which means a need for more food. And at the same time, it means cultivating salt-affected soil will become more common. However, selecting a right plant for cultivation in a saline condition and also at the same time effective soil and water management could be major challenge in the saline agriculture. So as we already mentioned, soil salinization is increasing worldwide due to the poor management of soil and water resources. These issues could be even more worse in the arid and semi-arid regions, and maybe in future many farmers forced to change their cropping strategy or even leave their lands. A study area in Iran had a long history of practicing saline agriculture and also growing salt-tolerant crop like pistachio, which due to its high resistance to salt, low water requirement, and at the same time a valuable product for export. The suitable EC for pistachio tree is less than 4 deciliter. However, it was also like it gives suitable product also in 8 deciliter. So using this high salt tolerance plant like pistachio seems good decision for saline agriculture, but it should be considered that soil and water degradation could accelerate due to the agricultural use of unsuitable lands. The use of saline soil and water for agricultural and also associated problems such as soil degradation in arid and semi-arid region had led to variety of research which focus on the saline agriculture and soil water resources management. This soil affected soil could be varied due to the physical and chemical characteristic of the soil and also depth of water table, leaching capacity, and salt distribution. Therefore, it's really important to monitor soil salinity, spatial temporal changes on the different scale, like what we did in the field scale. This is really important for maintenance of soil health and make right management decision. So basically, salinity study methods are like based on classical method, or we can call it traditional method, or like the traditional one that we go to the field, took samples, take them to the lab, which is really lab work, like with intense lab work and time and cost consuming. But also we can go through the new technique like remote sensing, proximal sensing, and also digital soil mapping. The, the method that we use in this study is digital soil mapping, which is based on a score pan model. You can see it in the right corner in the top. First, this model was introduced by McBaranty in 2003. Digital soil mapping was used to map salinity in this study. it connects the soil data that we collect from the field to the auxiliary data that we gather from the remote sensing data through the random forest model. Therefore, the goal of the study was using random forest model as a quick and reliable method for field-scale soil salinity mapping in central part of Iran. The study area was almost 135 hectare, and it was a field of pistachio trees. Mean annual temperature was 16.6 centigrade, and mean annual precipitation was 127 millimeter. Soil moisture and temperature regime were acidic and mesic. As we need some soil sample to connect them to the covariate during the digital soil mapping process, we took some sample from the field, which means at the beginning we went with the portable devices to the field, and we test the salinity variation across the field. Then we decided to do soil sampling in the grid design with 100-meter intervals, and then transfer those samples to the lab and measure electrical conductivity in them. Then after gathering all the soil data, which here they are measure electrical conductivity in the lab, and also preparing auxiliary data that we calculate them from remote sensing based spectral indices, which we use Landsat 8. And also we had some train attribute from digital elevation model, and then all of them were prepared in the 30-meter resolution. Then they were connected to the random forest model. Here you can see the produced spectral indices from Landsat 8, and also train attribute that we use. We use Saga for extract them from the digital elevation model. These auxiliary data can serve as a beneficial resources in scenario where fundamental soil data are limited. And then finally, we run the model, which was, as already told, random forest. It will build many decision tree in the parallel to connect measure soil salinity data and also auxiliary data. You can see that finally it produced salinity map for us. This model has a good ability to predict nonlinear association among categorical and numerical variable without any need of assumption about the distribution of the data. And the underlying actually principle of decision tree is that it dividing the data set into a smaller subset, and during each split, the algorithm selects the division that minimize entropy in the subset results. Data set splitting continues until achieving perfect classification of observation in each part of subset. And then this result that you can see is based on the classification tree that we use to produce of salinity map based on salinity classification, which defined by American Salinity Laboratory. And then I don't know if you can see, but the most part of the study area is red. But at the same time, whole part of the study area showed the electrical conductivity more than 4 decimeter, which means the whole area are in treatment of salinity. And this was completely in, this was also confirmed by our field visit that we had at the beginning. So if I want to summarize our results, I would say like approximately 33% of the study area was in the salinity class 4 to 8. Decimeter 20%, they fell in 8 to 16. 15% in 16 to 32, and we even had class greater than 32 decimeter. And also one of the other ability in random forest is like covariate importance can be fascinated, but by substitution, the original values of covariate with randomly one, which means by substitution, the covariate with the new one, it can increase It can show us the most important covariant because if we randomly substitute the covariate, if we took off the most important one, the error of the model will increase. In this study, the most important covariant based on this were the NDSI, TWI, CNBL, NDVI, and MSVI, which you can see from 5 important covariates that we have. 3 of them were from remote sensing. And also based on the tenfold cross validation, random forest predicted soil salinity map with the coefficient of determination value of 0.56. And also this map was inconsistent with our field observation. And finally, the digital nature of this salinity map allows us to update our information faster and in lower cost, and also in the different scales in the future. Thank you for your attention.
Yeah, thank you very much, Dr. Leila,
Welcome.
for your great presentation. And let's see if there are any questions. Please feel free to write your questions in the chat.
So I can see one that has the wide random forest model were used. Is there other model to mapping? Yes, so we can use different machine learning model, and then it depends on the on your goal first of your goal of the study. But recently, random forest for salinity was widely used, so we choose both random forest and also neural network. But I just show here the random forest one, so you can just try different model and see which one is working better. And I can see another question that is like asking why R square for random forest is quite low. What kind of hyperparameter tuning did you perform? I'm not sure about the second part of the question, but about the R square, I will say it's quite okay because the R square is based on the variate that we prepared. So in this study, we had covariate and also all covariate in 30-meter resolution, which means it's basically not really a high resolution for a small field. So if you have access to the high resolution covariate, you can increase your R square might increase as well. So for mapping, I use R, like software was R. It could be different like software, it doesn't matter, but it's basically code that you can produce like in both R and other software, coding software.
Okay. Let's see if there's any other questions. And have you tried support vector machine also?
No,
Which one
no.
results better for soil salinity? And have you used any index? Do you know if there's any index in this area for extracting, for detecting salinity automatically using index?
What kind of index do you mean? Do you mean salinity index?
Salinity index
Yes,
is
so I think I show in the slide, but it's really tiny. Maybe you couldn't see in the table. So basically, we produce 9 salinity indexes from the literature review. But of course, the formula can change based on the, based on the your area and your study area. But from these like 9 indices that we produced when we were looking at the most important covariant, they were not really helpful. So the most important one for us still was NDSI and the other one that I was mentioning, like SAVI, not those that we produce. And I will say it's because basically the satellite image that we got, the resolution wasn't really high. So it was like 30-meter. And for this small field, maybe it's not the best.
Okay, then random forest gives better result, yeah.
Yeah, it got better result, better than the neural network one, and also I think there was another one that we tried. I think we tried also like the normal method like kriging. So this one was the best one.
Okay, Lionel asks, what are the basics to make them model in order to trust the result?
That's actually a really good question, because when we are using digital soil mapping, if we really want to like trust the result, first of all, based on my experience, I will say it's really important that you have a good focus on your study area. That's why I was mentioning that we had a pre-visit to the per-study visit for field, and we know we know really the study area, because in many cases, when you are using many covariates, maybe this modeling ended you up to unrealistic results. But if you know the study area, and if you know like the covariate that you extracted, you can do correlation tests and just remove those like covariate that you think they are leading the model in the wrong way. I think in that way you can trust the data that you have by the end.
Yeah, thank you very much. Thank you
Welcome.
to both of our speakers and to all participants. With that, I would like to hand over to Action's chair, Dr. Kate, to close our last webinar. And this concludes our today's session. Thank you.
Thank you very much for participation in this webinar series, and also to Saeed for hosting and moderating this event. We hope to see you soon with the next series coming in the next year for Sustain as well. Thank you to our great speakers and to all the participants. It was great to be here as well. Thank you very much and have a good afternoon.